So how is your training ongoing? It's going good so far. Eco के मामले में like there is a eco. अच्छा. So this is probably the last lecture, or there are more lectures. The data analysis lectures will be continued, but as far as the VAT lab is concerned, this is going to be the lecture where today we'll try to discuss what are the different key elements where you need to put attention when you are setting up an NGS reaction. This is very, very critical because either it is ion torrent or it is Illumina. Every time you press the switch, okay, that is costing minimum 1.5 to 2 lakhs of rupees. And it's not that easy. If you remember, all the fighter pilots, when they are flying, say MiG or Mirage or whatever the planes are, the cost of the plane is written on their shoulders here. So just to make them realize what is, the responsibility that they are carrying over their shoulder. I mean, what would be the one cost of Rafale or say. Similarly, in molecular biology, as you go for the high-end instrumentations, specifically in genomics, it is not the capital cost that matters, it is also the running cost. Each time you press the button, it has a cost attached to it. I'm only considering the monitoring cost. Other than that, there is a resource cost, there is a cost of time, there is a cost of output, everything. And that is why it is most essential that how you set up the reaction as good as possible so that you end up losing the money, the time, the effort, the resources, everything. Okay. And for that today we would be discussing, there are a couple of aspects which are there. But before that, what are the different types of libraries? You must be aware that there are multiple libraries which are there in next generation sequencing. So say either it is a DNA, which is your starting material, or it is RNA. If it is RNA, then mostly you are going for transcriptome analysis. Okay. And in transcriptome, either you do the whole transcriptome. So in that there are now kits which have RNA as a starting material. So even the cDNA synthesis is a part of your library preparation. And again, the reverse transcriptase that you choose will depend on your application. Okay. So either you do the whole transcriptome sequencing, or there is a library for long non-coding RNA sequencing. Okay. Or you have a short non-coding, for example, miRNA library or siRNA library. Or you have a targeted sequencing for expression analysis, like you have it for DNA. In case of DNA, either you are going to study the genome or the epigenome. In epigenome, usually we do the histone modification studies or DNA methylation studies. Like you do chip sequencing for methylated DNA. Okay. To understand what are the epigenetic changes. You do sometimes chip sequencing to capture the rarest of the DNA change events that are taking place in your cell cultured populations or on your target cell libraries. Okay. Then in histone modification also you do the chromatin immunoprecipitation, that is again a chip-seq. And here it is in methylation, you do methylation deep sequencing method. So for that you need to understand. And in genome, there are multiple libraries. The simplest one is a whole genome sequencing where you are trying to decipher the whole genome. Again, in whole genome, it could be de novo sequencing or it could be reference based. In cases of bacteria where the sequence is mostly known or the nearby sequences are known, sometimes you end up doing reference sequencing. In that, you don't need that much of a coverage. For example, you know that the culture is Bacillus subtilis, but a strain is different, which is showing some different characteristics which are of interest to you. So in that case, you need not to go for the de novo sequencing. You can also go for reference sequencing, where with a less coverage also your purpose or your job is served. Another is a metagenome sequencing. And all of you know that the AMR training, excuse me, what you would be studying is that we have a custom AMR panel, right? And that AMR panel is targeting different antimicrobial resistant genes which we are trying to capture from different sources. It could be a fecal poultry sample, or could be a soil sample, or could be a human that is handling a poultry sample. So that is where you are doing targeted metagenomics. You can also do the whole metagenome, the whole shotgun. That means whatever community is present, you are just extracting DNA, and that DNA you are subjecting it to sequencing. Okay. But in some cases, for example, only you want to understand the microbial diversity present in that particular metagenome sample. So what do? You do 16S amplification. And that 16S amplification you subject to whole genome sequencing. So that is called as targeted metagenomics. I mean, in shotgun sequencing also. The whole exome sequencing. This is very important for clinical samples as far as human clinical samples are concerned. Usually, you want to study only those regions which are expressing into some peptide. Okay, so that is where you study the exomes. Now, again, it could be the whole exome or it could be the targeted exome. For example, in GBRC, we are studying whole exomes to understand what are the risk of hereditary breast and ovarian cancer, okay, which are already predisposed. Similarly, we have a custom muscular dystrophy panel in which it is a 28 gene panel which can differentiate 29 different type of muscular dystrophies in a target population. So it could be the whole exome sequencing or a targeted sequencing, or it could be the whole genome sequencing and then metagenome sequencing. So I will not go into the entire flow, but typically you know that once you do the sequencing, then here you do the mapping with the reference genome in case of exome and targeted, and then you identify different SNPs, index, copy number variation, or the structural variations which are present. This is more for clinical application. You also do the whole exome sequencing, do assembly annotation, and you do the putative gene identification for any other samples. For example, and same is with the whole genome sequencing. If it is a de novo sequencing, again, you try to understand, for example, we are trying to do the lion genome sequence. So that sequence was not published previously. So you know that it is a eukaryote with a C value, you estimate the total genome size. So what is a human genome size? Anybody can tell me what is a typical human genome size? Around 3.3 GB. Okay, and if you are going into discovery mode, you take minimum 100x coverage of that particular sample, which will give you around 330 GB of data, gigabase pairs of data. Similarly, in metagenome also, you can understand the diversity, or you can understand the genes which are active in a particular ecosystem. For example, we are running couple of projects where we are trying to understand the diversity, not only limited to the structure, but the functional diversity, which are the active genes within that metagenome. That is a meta transcriptome. So why I am giving you this glimpse is to understand that whenever you are doing your NGS, you should know what is the objective. And based on your objective, you would select the methodology to go about it. Okay. So this is the glimpse of that. Now here you would have seen, or probably in AU you have seen MySeq. Here we have MySeq as well as ion torrent both. So this is just a broad picture of, once you have your target, whether it is a DNA, RNA or cDNA, that you subject it to library preparation. That library preparation for ion torrent typically involves enzymatic digestion. So usually, your library is a whole stretch of target nucleotide sequence. If it is a DNA, it is a whole genome. Okay. Then you want to cut them into desired pieces. Now, these desired pieces are basically based on what? Your sequencing chemistry limitations. If your sequencing chemistry is 600 base pairs, that means the machine can read up to 600 base pairs, then you want to cut your desired library in that range. Okay, so it's like a jigsaw puzzle. You have got a whole picture, but you are cutting it into 500 or 600 base of size. Now that cutting can take place in two ways. One is a mechanical shearing, which mostly involves ultrasonication. Another one is enzymatic. So you use enzymes and then you make a specific size out of it. And then you don't want any other size. You only want that particular size. So then you do library enrichment and then preparation. In case of MySeq, the tegmentation process you would have seen, which comes into the picture. So basically after that, what happens is that respective adapter ligation takes place. And along with that, sometimes you add barcodes also, because you want to add multiplexing into the picture. You want to add more than one samples in one go. So you add different barcodes just to identify that this read came from this sample particularly. After that, once the library is done, then it is subjected to clonal amplification. Here in Illumina, you would have set up yesterday the emulsion PCR, right? In ion torrent. Emulsion PCR is basically you are making an emulsion where in the bead, your library gets attached with the respective barcodes and it gets amplified. So all these technologies, either it is Illumina or ion torrent, they are basically subjected out of PCR. So one thing you have to keep always in mind, that the PCR biases are always a problem whenever you are doing next generation sequencing. Okay. Here, in case of Illumina, you know that the bridge amplification, how does it take place, right? So there is a strand which gets attached to, this is a Illumina chip in which it gets attached and the primer does sequencing. The bridge formation takes place. Again, it gets repeat sequencing, that is a two-way sequencing, and then you have the both the strands over here, and then it is subjected to amplification. So here it is detecting at the end of the day fluorescence, and here it is detecting a change in a pH. With every nucleotide added, there is a release of hydrogen ion, and that is detected in Illumina. So these are the broader steps what are there, but what are the key checkpoints where you are bound to make mistakes, and those small mistakes can result into failure of the entire run. So we'll just try to run over them, couple of them. Okay. So the first thing in the library preparation is that to confirm and verify your starting material. This is the most common mistake. Whenever we receive samples, I would not like to name an organization, but even from a reputed institutes, we used to get ChipSeq. And in chip sequencing, they prepare the library, because the chromatin immunohisto precipitation technique they used to do, they used to send us the library. We would just subject it to sequencing here. Library preparation was not done by our team. In that case, they claim that you have contaminated our library with, some other sources. And then finally, we found out that it was already present in the library which was sent to us. But all this is lot of, back and forth once you are stuck. So you want to make sure that whatever, even if it is a bacteria, sometimes you get is a lone growth which is sent to you for sequencing. And unless you know it is an isolated colony, what you are sequencing could be completely different culture, and you will only get to know once you get your sequences and do blast and do the identification. So this is very important. You have to also deploy some microscopic methods. If it is a cell culture base, suppose you want to do virus sequencing, which we are doing here. So in that case also you need to first see whether the sample is pure or not, plaque formation is uniform or not. And based on that, sometimes, when your target organism is known, we also used to run a capillary sequencing to confirm the purity of the genomic DNA. For example, if it is bacteria, simply you run 16S to get to know whether the culture is pure or not, the DNA is pure or not before subjecting it to whole genome sequencing. So, the most important step begins with isolation of nucleic acid. If you are looking at a DNA, so this is how you run it first on the gel. The first step you will do is to run your whole DNA into a gel. And by looking at the gel image itself, you can understand, what are the issues in your sample, along with the fluorimetric method. So here, as you can see, that there is also a contamination of residual RNA. So probably you can do the RNase treatment and again go back, check on a gel, look at the ratios, and then unless you are satisfied with your isolation techniques, you need not to proceed further. In some cases, the DNA is degraded. Suppose the sample is too old and your DNA from FFP tissues when you are trying to look for a cancer or some other genes. So you need to then use some specific kits, Or sometimes you are sequencing from bones or even the dry samples in which there are very scarce chances of getting DNA. And in that case, you have to first optimize DNA extraction. Unless you are sure that the DNA is a good enough DNA, or at least we can say a PCR amplifiable DNA, then only you can proceed further as far as NGS is concerned. Another method to check is the quality and quantity of DNA, which is mainly either fluorometric or it is a UV visible spectrophotometric method. So that this is a nanodrop. Now this was initially used, you have to just take your sample in a dip, put it in a nanodrop, it will tell you the ratio as well as concentration, everything. One of the major drawback over here is, the carryover contamination. And any person who is running next generation sequencing, or even for that matter of fact, a PCR, they need to remember that carryover contamination is a major issue in any sequencing lab. Because every day you are dealing with DNA, and that is, very sturdy material. It remains in your environment. And there are very high chances that it can contaminate your libraries. You cannot differentiate whether it was a carryover contamination or your own DNA. And for that, there are some solutions like DNA out or DNA zap, RNA out. There are certain chemicals which you need to regularly use in your PCR area as well as sequencing area cleanup to remove the carryover DNA contamination. Then this is again a Kaya Gen instrument, that is Kaya Expert. that also does a chip-based identification of concentration and the quality. This is fluorescence based, which is a qubit fluorometer. Currently, we are using qubit 4. So here, the low accuracy and sensitivity is there. But when you are going for fluorescence based detection, it is highly accurate and it is sensitive. So far as DNA quality is concerned, if it is impure, then it will hinder your library preparation process. Okay. Because if it is, there are residual salts, it will interfere with the shearing process in enzymatic digestion. So these things you need to keep in mind. So the pre-analytical QC. So usually when you are looking that your DNA is not migrating in the gel, either it could be protein contamination or the salts also could be higher. And in that case, if you try to dilute your samples, then you can get to know. Now, DNA integrity also can be checked on a TEP station. So this is a typical image. As you can see that these are 3 different samples which are added. So the blue one is a poor quality sample. As you can see that the DIN value is also very less, around 2.3. The green trace is again a sample with a 5.6 score, and the orange is a good quality DNA, as you can see, which has a DIN score of around 6.5. So these are the various methods, combination of electrophoresis, fluorometric, or kind of combination of both. So you would have seen here one more instrument which is Kaya Expert, which is also an automated electrophoresis, which will also give you a good picture of how your DNA is there. In qubit, when you are doing estimation, then you have to remember, that all the reagents must be at a room temperature. Then you have to also protect the reagents and the working solutions from light, because it is a fluorometric, and the light exposure will reduce. The best way to handle NGS every time is for the high-end consumables, you must prepare ELI codes. The day your solution comes or you are opening it for the first time, you have to make them in ELI codes so that you don't end up every time thawing and freezing, the same bottle. So you know that in one run or maybe for over a month, what are your requirements? You have to ELI coat your samples like that. Even that is the case for primers. In GBRC, we have a process where we have a primer repository. So anytime anybody is ordering a probe or primer, it is not directly handed over to them. No day. Because there are different people. If you're talking about 16S application, probably there are 20-25 fellows who would be using the same primer. And anybody can contaminate the primer, and you will not get to know unless you know the final results. Okay. So best thing is to do ELI coating. Even for real-time PCR, we are doing a lot of quantification work over here. You get your samples in a bulk, like 10 ml master mix is there. And per reaction you are going to use is only 10 microliter. So you should not every time, thaw and freeze the solution. You have to put it in ELI codes. Primers also, dilution is very, very essential. You can end up diluting your stock in a wrong fashion, which can spoil your entire, downstream processes. So dilution is also done by experts, and then only the working stock, which is around 10 picomole, is only issued to individuals for their regular use. In NGS, like we have our own barcoded primers of 96 sets, and we had faced this issue. All these issues you will learn by experience. Once you commit a mistake and then you spend your time, hell lot of a time troubleshooting what went wrong, and then you will realize like 16S barcoded primers were contaminated here once upon a time. And then, whatever you may sequence, you would get whatever, the contaminant is there, you would get amplified. So these things are very, very small, but very, very critical. Sometimes you may feel, it's nothing like that. We used to joke when we used to run for NGS that last time when you performed a reaction and suppose by chance you jumped twice and you got good results, so you must jump twice. So the reason being is, it is more of how you follow the process. Jumping indicates that exactly following what is written. Sometimes, we get casual when we start handling more and more number of samples. We get casual. And every time you get casual about this, you end up making mistakes. So that's the point that I am trying to highlight. So, so that's why, follow the recommended incubation time. So sometimes, people used to think, oh, 5 minutes, 5 minutes, okay, I'll see in my watch. And then you forget while you are doing some other stuff. So always use stopwatches in a critical incubation time where you are exactly looking at the timing. In nanodrop, as I told you, that the carryover contamination may increase or decrease the reading. So carryover contamination could either your sample is less concentrated, but because of the contamination DNA already present, it will show you higher quantity. You will end up taking accordingly, because in any enzymatic shearing or in mechanical shearing, you have to take a specific prescribed amount of DNA. And that precision is coming from the quantification steps. So these are the possible points where it can go wrong. Then blanking reagents. These are very, very small but very essential things, because in qubit you do a blank. Okay. Similarly, in nanodrop also, the buffer in which your DNA is dissolved, you use it as a blank. So try to use the same pH and the ionic strength. Sometimes what happens, your master buffer stock is 10x. Okay. And your diluted DNA, whatever it is, finally dissolved it is in 1x. But when it comes to taking a blank, we don't have that empty 10x. And we sometimes feel आलस कि अभी इसको dilute करने के लिए कहाँ फिर से 1:10 बनाएंगे, यही ले लो ना, क्या फर्क पड़ता है, इसमें तो कुछ भी नहीं है ना. So that can also end up getting you the wrong results. Okay, so now assume that everything went well and you have got your DNA or RNA, whatever was the starting material. For RNA, I didn't mention over here, but once you do RNA extraction, then you have to either run it on a gel and usually check the ratio of 18 to 28S, and if it comes out in a range, that is okay. If you are taking it for transcriptome, you can as well run a bioanalyzer and check the RIN value of your sample in the range of 1 to 2, and 10 is the least degraded, but that's an ideal. Ideally, if you are getting a RIN value above 5 or 5.5, you can proceed that sample for transcriptome analysis. Fragmentation in ion torrent is done either enzymatic or the physical. As I told you, physical or the mechanical rather is the right word. That is basically done on Covaris or any other ultrasonicators that you have. And enzymatic is done with the help of different nucleases. In Illumina, it is a tegmentation process, which is again based on the time. Now, when you are doing a mechanical shearing, so first you need to understand the make and model of your ultrasonicator, what frequency exactly whether it is a probe based or a non-probe based version that you are using. Water level is very essential whenever you are using ultrasonicator for mechanical shearing of your DNA. There should be no bubbles in the assay tube, because otherwise whatever frequency you may be thinking that you are giving, but the actual frequency getting transmitted would be different. In case of ion torrent, you know, you also have to do a end repair, 3' and a tailing, if you are using the mechanical shearing, and the sample should reach 4 degree, and you have to hold it basically for 5 to 10 minutes before adding adapters and enzyme makes. Similarly, in enzymatic shearing also, you have to use ice, which is very, very critical. Otherwise, if you think that you just took out the enzyme mix and the sample is not kept on ice, you know, at 25 degree also there would be some shearing which will begin. And timing and the sequence of addition of the stop buffer is also important. If you have around, so this is important where you are handling more than one libraries at a time. And that would be the case for 16S and all other applications where you have to do either 12 or 24 libraries at the same time. So stop buffer आप उल्टा डालना चालू करोगे, so पहले ट्यूब में टाइम ज्यादा चला जाएगा, लास्ट ट्यूब को सबसे कम टाइम मिलेगा. So all these things are, that's why, very small but essential. Now this is a comparison of the mechanical and enzymatic fragmentation method. So as you can see here, the blue trace is basically a sample treated with an enzyme, and the orange one is the Covaris, or you can say ultrasonicator based method. And as you can see the range, so mostly the preferred method for fragmentation is an enzyme based method, where, because you know, with a proper incubation you end up getting almost narrow peak of your library fragments rather than the broader ones. And this is a comparison of an enzymatic fragmentation incubation time using Agilent high sensitivity D1000 screen tape. And here the graph is showing that the increasing the fragmentation time results in the gradually decreasing peak size corresponding to the reduced yield. So as you can see that the time is very, very essential, and you need to manage this. After your library is taken out in a proper size, the next step is the adapter ligation and library amplification. Why you do library amplification? Can anybody tell me in NGS? Why you do library amplification?
Always the target so that we get the clear and distinct image of sequence.
So basically to increase your library concentration, that's the whole objective. So in this, you have to take a proper master mix and adapter concentration, because if you take a higher, so this is ideally 1:1. One fragment of your DNA should bind with one adapter. That has to be 1:1. Now what happens here, if you do not take proper, adapter is higher and your target is less, what will happen? Adapters will do the self-dimerization, and then after that you do not have any mechanism to rule it out unless you do sequencing. Similarly, if you take less adapters and more library, what will happen? You will end up using some library fragments which are not properly ligated with your adapters. So this concentration is very, very critical. Okay, taking 1:1 concentration for your library, I mean the sheared fragments of and adapters. After the incubation time, you know, your adapter and library, you do a incubation, immediately You have to take it for a purification, because whatever leftover adapters are there, if you increase even the incubation time, those adapters can do the self adapter ligation. And then based on the library concentration, so we will then take this library after adapter ligation is done, to subject it to library amplification. Now this is again very critical step. Hypothetically, you are assuming that your each library different DNA is getting amplified, okay, in equal proportion. But it will never be a case, because it is a PCR, and PCR can always have biases. So ideally, sometimes you do say 8 cycles to get your libraries in a proper concentration. Then again you go back and check, and then you think that okay, my library concentration is still not appropriate to take it further. You may increase 1 or 2 or 3 cycles, which will again end up having more and more polyclonal libraries, So it would be only a one fragment which would have a higher yield. Your ideal condition is that what you have done is the 3.3 GB, which is a human genome. Assume that you have cut it into 500 base pairs or 600 base pairs, and you want representation of each that 600 base pairs in your library, which is amplified uniformly and then sequenced so as to get the final coverage as desired. Otherwise, there will be a bias. Some regions are sequenced more, some regions are sequenced less. So you need to actually reach to a optimum time where your library is getting amplified properly. So this is based on your library concentration. Every your protocol is actually giving you whether how many cycles you need to proceed, and for that you should take the lowest possible cycles to avoid the duplication in reads. So this is again a very critical step. So here again, if you are looking at a library size, once you have your library prepared and amplified, you do the quality control of library. And for that also there are different steps involved. So this is again on Agilent screen tape assay. And here, as you can see, this is your ideal library size, and these are the markers. So the size selection is again very important. In some cases, when you do enzymatic shearing or a mechanical shearing, this is how your DNA will look like. Okay, and this is called as e-gel. So here, while your sample is running, you can look at your sample real time. And this is a marker. So suppose, this is 100 base pair, 100, 200, 300, 400, 500 and 600 base pair has just entered. Whatever DNA is there, that much you are enriching. So this is one way of purification, which is a e-gel based, where you are visually looking at it. Another is using the beads. So these are Pure XP beads, which are again proprietary ones, and these are magnetic based beads. So usually, they have labels which capture your DNA. So these beads are very sticky, and that's why you have to make sure that first of all it is thawed properly. Again, beads also you need to make aliquots. Otherwise, every time you are using it for different library, you will end up contaminating beads itself, which can also add a lot of difficulties in the final sequencing. And vertexed properly, because they are very sticky. So you have to thoroughly vertex them before just taking it. So usually if you would have seen here or even in Anand, Those who are doing the bead purification along with the magnetic bead stand, they have kept the vertex also just adjacent to them. And every time you just vertex and then take it. Okay. The 70% ethanol needs to be prepared fresh every time, every day rather. Because ethanol can evaporate and you think it is a 70%, actually it may not be 70%. And that's why it is always recommended to use it fresh. Now, when you have to do, purification, so there are some steps where your library is binding to beads and you have to discard supernatant. And once you do the, buffers to release your library in the solution. So these are also very small, but people end up making silly mistakes that where to discard supernatant and where to, discard the pellet. So sometimes, you have discarded your solution as a supernatant, you end up again losing everything. It is again very important, and this again comes with a time only, that when it is dry. And this is, believe me, very personal judgment. Even if I tell you 1 minute is appropriate or 30 seconds is appropriate, it won't matter there. You have to look at the palette. As you can see, there is a little shine over here. So that means palette is still wet. Here you see there are cracks. You can see in the palette, that means it is over dried. And this one is a matte dry palette without crack, which is the optimum time. So you need to again make sure that when they say dry, it should not be wet. Here what will happen? Your concentration may change. And here if it is over dried, that basically will reduce the efficiency in elution buffer when you are eluting your DNA, and you may end up actually because of overdrive. So, library purification, as I said, you can do either bead based, which we discussed, or you can do QC of the library, either again using qubit only for concentration purpose. This is a typical image of Kaya Excel where you also get to see the size, and this is real-time PCR for library quantification, and this is digital droplet digital PCR, or we also have another version of digital PCR for library quantification. This is very, very critical step. Okay, because if you take your library concentrations wrong, then your entire reaction at the end will not give you desired results. It will fail. Very, very critical. So typically in GBRC, this is the step where we give most of our focus. What is the library concentration? We do it using bioanalyzer, and then followed by that we do either with the qPCR, real-time PCR, or with the digital PCR to estimate the exact library concentration and then decide the dilution factor based on it. For dilution factor also, it is always easy that you should have one Excel sheet, and rather than calculating it manually, that whatever formula you have fed in based on your dilution factor and the final library concentration that you have to take, it is always advisable to use that standard Excel sheet, because sometimes 1 0 here or there would end up entirely changing the library concentration tenfold more or tenfold less based on the mistake that you would have committed. So for library quantification, as I told you, quantitative PCR you can do. So these are adapter specific primers. And why this method is more reliable? Because it counts only those piece of library which is bound to your adapter. And then also there is a standard library which is provided with the kit, which you dilute it serially in a different concentration, and against that standard curve, you can actually quantify your sample. And sample also typically needs to be taken two dilutions based on the concentration that you get from qubit. Okay, so you take two dilutions, so you are cross checking it twice whether your library actually quantifies it to what is actually corresponding to. Then fluorometric assay, as I told you, and then bead based normalization, sometimes this is also followed, and then the bioanalyzer. Typically in GBRC we use combination of three, that is a qubit fluorometer, the bioanalyzer, and the quantification either using digital PCR or qPCR. First day I think you have seen the library quantification using both the techniques, right? So that is a standard practice to be followed. Now in quantification also, bioanalyzer has a high accuracy and you can depend on dilution because it also gives you a picomole concentration of your library that you are trying to run, and it is recommended only for the quality control. But again, here you can not differentiate the adductor ligated and non-adductor ligated because it is just a size based method. Fluorimetric double-stranded DNA assay. This specifically detects the double-stranded DNA, but it can again not discriminate between the complete and incomplete libraries. qPCR is a specific measure and it will give you a very sensitive, accurate concentration. It also UV spectrophotometric based method. Ideally, you should not do it for library quantification. For DNA, it is okay. Should avoid using because here there could be a non-specific detection also and contamination also. You cannot discriminate over here. Uh, so this is how you have seen the bioanalyzer yourself. These are different, wells where you pour the gel and like a normal electrophoresis along with the size marker, it runs and it gives you the library picture. There are three different chips, DNA 1000, DNA 7500 and high sensitivity DNA chip. There is a lower size marker and an upper size marker, and based on that your sample peak is visualized and analyzed both in terms of size as well as concentration. But it cannot differentiate. Sometimes you can see, there are adductor contaminations. If it is there, it can be visualized in bioanalyzer. So these are the base pair ranges and the quantification ranges of the each assay kit. You have to choose it based on your application. So once your library is ready, calculated by the intensity of the highest, which is divided by intensity of highest plus second highest signal. Okay. So typically you spike in 5%, but if you have a less heterogeneous library, which I said as your 16S library, it will not be that heterogeneous. Okay. In that case, you have to take almost as high as 10% fixed control to neutralize your reads in the sequence. This is for ion torrent. Again, when you are going for amplicon based library, what are the do's and don'ts? Like if there is a poor purification, then you can purify with the more number of pure beads volume. I am not going into detail of this, but this also is as good as you have to evaluate what went wrong where. This is a ion torrent run. So here also they provide you control ISP beads, which you have to spike with every run to ensure that your sequencing consumables are working okay or not, your machine algorithm has worked properly or not, just to check that. So here you can see there is a 91% loading. That means loading is very good. Okay, it is uniform. 100% of the library is enriched, but polyclonal is 24%. And that's why the final library concentration is 88% only. So what is a polyclonal? You want each clone to be represented equally. But when your library becomes polyclonal, that means some of the clones are getting represented more in a particular set of library that you are preparing. So this is the example of polyclonal and monoclonal, because you want each typical bubble to have one type of library fragment. And to overcome that, you also again need to check the adapter dimer contamination or the high amount of the library which is loaded in emulsion PCR. What will happen if the high amount of library is loaded? There are some fragments of your library which have a higher PCR affinity. They will end up getting amplified more. And then you can see, the graph like this. Finally, it will give you a read length map of all the reads which are generated. So suppose your library is a 350 base pair and you observe a graph like this. So again, there could be issue in the emulsion PCR. You may have the kit which is an expired, and that is why your library is not fully covered. You were expecting that it will go up to 350 base pair, but it ended in 200 base pair only. Or sometimes your quality of TCAG, consensus key MERS are very less, then again it indicates that the sequencing reagent kit might have expired. So these are the issues which I talked about it, that coverage dropouts. Then small sized amplicons or the high GC content in your library. And this already we discussed about the ambiguous clustering. So low diversity library or the wrong quantification can also result into ambiguous clustering, and insufficient purity of your library also can result into this. These are the references from which this is taken. And then if there are any questions, we will take that, or else I think everybody is feeling hungry. Which one is better? So yeah, that issue, I thought it would come. And because we are using it both, you need to understand what is your application. Okay. If your application is a discovery, you are doing something de novo, then Illumina is better because it gives you a higher Q score. But if it is not a discovery and you are suppose looking at only sequencing the same, for example, the best example is Ampli-Seq kits for AMR also. We have checked. I think Twinkle has a data where we run the same libraries on Illumina and Iron Torrent. So one is giving you Q20 result, the another one is giving you Q30 results. Even there was a difference in output. In Iron Torrent, I think we had only around 50,000 reads per sample. So that also you need to determine that what is your size of the library and multiplied it with the expected coverage. That will give you ki this much minimum reads are required for sequencing in order to get a particular X coverage, which is a publicly or in publication acceptable coverage range. In both of the same library, we got the same results, almost similar results, identical. So that's what I'm saying. Again, it depends on application, which application you are targeting. And based on that, you can choose the platform. Any other question?
What is that entry parent you take?
So end repair is basically when you are mechanically shearing, no, it is a double-stranded DNA and it will not be shared homogeneously. So you have to repair the ends because then at the end of it you have to take it for ligation. So you have to undertake that. There is a kit that comes. It used to come with Illumina end repair kit or something in Iron Torrent. So there is a polymerase which will fill up the ends and make it a uniform double strand.
Because in physical shearing, all the DNA fragments may not have the blunt end, exactly blunt end. So you have to make the ends blunt for the adapted ligation. Because you get the end with the polymerase and nucleotides.
But mechanical shearing is never a preferred method for library preparation. The reason being you require a higher input even. In mechanical shearing, I mean, you have to take sometimes microgram of your libraries in order the input DNA for shearing. And in enzymatic, with the less concentration of your input DNA also, you can make good library. Okay, if there are no questions, then thank you for being so patient because it's almost a lunch time, so everybody must be feeling hungry. And meanwhile, also, if you have any questions related or unrelated, you can comment this.
ત્યાં મોકલાઈ દીધું છે.
મોકલાઈ દીધું?